So early this year, I had my PhD progress report meeting, and I was asked, Stijn, it's going very well, your PhD, but we are now very curious in this final year that you're heading into. What will you do to increase the impact of your work, to take your project to the next level? What do you add to the scientific field? What is it that is the Stijn signature? Welcome to the show. I hope that you will learn something. Welcome to the show. I hope that you will enjoy yourselves. We'll talk about my favorite cell. My one and only true love. We'll talk about my favorite cell. The endothelial cell. So, before I begin, some household announcements maybe. For the people that are at home, that I'm not talking to right now at all. For the people that are at home, I have no idea how this will come across. Apologies, so next time be here. To the people here, I have no idea what I'm doing. So some things will work, some things will fall completely flat, but alas, such is life. And I think, as you might notice, I'm also quite nervous. So let's just head into my safe space, that is the endothelial cell. I think we all know that endothelial cells cover all the blood vessels in our bodies. And as such have functions in making sure the blood flow is regulated, allowing different immune cells to pass through our blood vessels. But although the endothelial cell is a single cell type throughout our body, it's actually quite different. And those differences are based on where our endothelial cells reside. It's based on the microenvironment the cells are in. So things like flow, things like the basement membrane they're on, things like the shape and size of vessels all influence the phenotype of our endothelial cells. And why, if you then translate that, that means that the endothelial cells in my brain are different than the ones in my heart and different than the ones in my lungs. And even within an organ we have different types of endothelial cells. And the idea is that, why is that important, or what is our selling point here, is we know that there are site-specific, organ-specific vascular pathologies. So could it be that this endothelial heterogeneity underlies these diseases? Now, to study this heterogeneity, we have two main camps. One is the mouse transcriptomic side, who do single-cell transcriptomics in mice to determine this heterogeneity. And the other one is the in vitro human EC culturing systems, where they test functional assays, of which I'm one of them as well. Now the mice side always says you can't do in vitro functional assays because a cell, an endothelial cell, will adapt to its microenvironment, so it will also adapt to its in vitro microenvironment. But that does mean there's still, that does mean that still a lot of studies actually on so many different endothelial cells are done in vitro. So that also means there's some gaps in this if you look at this. One, transcriptomics is not proteomics. The translation is often quite poor. Moreover, we are not mice, we are humans, so we miss a human factor. And we also don't really know how the proteome changes in these in vitro systems. Is that really that different, or is it all the same? So that's where we set out to do. And this is our workflow. So we acquired endothelial cells from all kinds of different organs that you can see on the screen here, commercially acquired, brain, aorta, cardiac, lung. and also included some of our favorite in vitro models, which are the BOEX, derived from the blood, and the HUVEX. We also did some stimulations to see if the inflammation response was any different. And then we do proteomics because we are the proteomics group. So to set the scene, the baseline, there are endothelial cells. So this is all what we see here in green is von Winold factor, which is endothelial cell marker, and then the VCAT here is there in the cell chips. So that's nice, at least, that they are endothelial cells. This is our amount of proteins, so we reach just shy of 8,000 proteins per sample, which is quite the proteomic depth. So let's show you the first result then. The first result is actually indeed that these cells are very similar. So this is a heat map of the Pearson correlation, and it tells us something about the overlap between proteomic datasets or datasets in general. And what we can see, or what I can tell you, is that most of these are actually over 0.96, which is a very high correlation. And the most different ones are the HUVEX and the BOEX. Now this is overlap. You can also look at the differences in PCAs. So this is a bit more difficult. So PCA1 of PCA2 and PCA1 of PC3. And a PCA tells something about the variance between data sets. And PC2 very nicely separates our interferon gamma response. So those are that's actually quite a nice separation. But this PC3, you can already see some differences. So yes, there is some organ separation, some BOEX, some livers, cardiacs, brains, kidneys, and the HUVEX. But we can also see that for some cases, and for a lot of cases actually, there's this massive donor-to-donor variation. Especially for these lungs and here's the aortas, meaning that the donor variation is sometimes higher, more than even the organ variation. Which means we can't do classical grouping statistical tests. And that's why we went to do the weighted correlation network analysis. And this is an outcome of it, a visualization of the outcome. And although I think it looks very pretty and would not be misplaced on the walls here at Sanquin, it's quite difficult to understand. And just as a disclaimer, I did not actually, this is a Photoshop, I did not remove the piece of art that is already there. So, it is quite difficult, this analysis. So I thought, how to make this easier? Well, I don't know. Let's just write it into a song to explain it, which I don't think will make it any easier, but for the very least, it might be a bit more enjoyable. And I do need someone to click for me. I need to walk more here. Oh yeah. I told you to come. No. Can you click for me? So every time, I think it's very clear. I will look at you. Whenever I say step 1, then you click, step 2, then you click. Yeah? It's part of the act then. Oh. All right. So this is how you do the. This is how you do that. WGC analysis. WGC analysis. Step 1. Step 1. No, that was step 1. Step 1. Put all your proteins in a line. Step 2. Step 2, now determine the pattern of regulation that these proteins follow. In what samples do they go up, in what samples do they go down? Step 3. Now group the proteins based on the regulation. So proteins that follow the same pattern, we put in the same group that we call a module. Wow. Step 4. Now, per group or module, determine the molecular function, the biological process, or cellular component that these proteins are enriched for. So that means that now we have on the one hand, we have the biological regulation of how our samples are regulated, and on the other hand, which biological processes these are. So I think that was quite easy, I think. So this is how you do the WGC analysis. WGC analysis. WGC analysis. WGC the WGC analysis. So what do we learn from this WGCN analysis? Oh, I have to talk here, I'm so sorry. So what do we learn from the DCNA analysis? Well, let's go through it step by step. So the first 2 or 3 modules very nicely describe the main separation in this network. M1, M2, M3, though, these are the biggest one, and they say something about the separation. So all the molecules on this side sort of enrich for proliferation processes, DNA binding, chromatin organization, ribosomes. While all the things on the other side seem to enrich more for extracellular matrix, actin filaments, cell morphology, and we term that more differentiation. Now, in this more differentiation cluster, there's cluster 6, which has a lot of enrichments, and this is a slightly different one, the old one out, because this is our interferon gamma clusters, and I make it a bit bigger for you. So what we can see is that the interferon gamma response is indeed highly similar across all of these cell types, similarly regulated, similarly upregulated. So this response is similar in our endothelial cells, which begs the question, is it similar in our endothelial cells only? Or is it potentially similar in all of our cells? Or is it potentially that the endothelial cell is similar to other immune cells, which have very similar interferon gamma responses? But the main message is, regardless of what endothelial cell you are, in vitro, your interferon gamma response is very similar. And then we have also some more organ-specific clusters. So if you look at M37 there, on the right, that's specifically in liver, 2 out of 3, enriching for extracellular matrix. And of course, the highest here is TIMP1, so this follows the pattern of this cluster the most. And that's indeed a protein found in the extracellular matrix of the liver, so that's nice. And then we have one here, M36, that's also a pretty interesting one. So that enriches for inhibitors of proliferation and tight junctions, so junctions between endothelial cells. And that's actually uniquely downregulated in HUVEX, which is one of our in vitro models. So the WGC analysis cannot just be used to discern differences in cellular processes or samples, but we can also look at it from a more protein-specific standpoint. Because all proteins that are similarly regulated, as long as your dataset is big enough and has enough variation, are often also in the same process or interactors of each other. And I'll show you an example. Well, we show a couple of examples, but today I'll show you the von Helmont factor interactor network. So if you have von Helmont factor, super important in endothelial cells, important hemostasis, platelets adhere to it when you have a bleeding. And if you look at the pattern, so that's A, there's a generic pattern, so slightly higher in boax, a bit lower in the brain. But it enriches for actin binding process, actin binding, which makes sense because von Helmont factor and is in viable palladic bodies, and these are trafficked, bound to actin filaments in the steady state. So now if we look at the proteins that are in this von Helmont cluster, and then we look at the proteins which correlate most to von Helmont factor itself, we see some things. So that are these. And what is very nice, if you look at the top 5, or the top 4 in this case, those proteins are all very well known, very well described interactors of von Helmont factor. So Rep3D, Myo5C, LIPS1, we all know them. But there's also some, so I show you these, that have a similar interaction score, but we do not necessarily know what their relation is to von Helmont factor. So in this case, through a completely data-based, data-generated, correlation-based approach, we can actually find putative, novel, potential interactors. And then we work together with some other groups, for example, with Erasmus, where we actually try to pinpoint some of them. Oh, that already brings you to the conclusions. I'll have a sip of water, I think, before I start singing again. So these are the conclusions. This is of course the most serious part. I think in any presentation, this is the only thing you need to remember. So I won't be using the guitar. These are the conclusions. These are the conclusions. These are the conclusions. So you better pay attention. So what have you seen? Well, our endothelial cells are very different, but they are also very much the same in vitro. Donor-to-donor variation is similar to organ specificity in some cases. That doesn't really work. The main axis of regulation is the one of proliferation versus differentiation. And the interferon gamma response is highly conserved across endothelial cells. Database generation leads to biological indication. Well, von Helmont factor, if you get here networks, So these were the conclusions. These were the conclusions. These were the conclusions. So I hope you have been paying attention. And no, I cannot create a drug out of this study. And no, I cannot help a patient to go to industry. And no, I cannot valorize it according to the Sanquin for life business strategy. But if I would have wanted to make money, I would have worked in a company. Science can be based on pure curiosity, on the human need to know. I feel sometimes we seem to forget this and only focus on the money, paper, abstract, poster, award, travel, prize, I don't know, first authorship, going, getting a certain number of papers, and in general just selling our science in such a way that it gets sold. So forget your presentations, your meetings and your papers, your supervisors, your deadlines and your conferences. Just go out there, do science, have some fun. I think that's originally how it was meant to be done. Before I round down, of course I have to say thank you to all the people here on the slide for their scientific input, but also the input outside. I think that's very important. Also thanks to my supervisor, Mark and Arjan, supervisors who said yes when I said I wanted to do this stupid idea. Thank you. And then we have to close down, of course, on where I started, which was the Stijn signature. How do you increase the impact of your work? How do you take it to the next level? How do you add something to this scientific field? Yeah. I don't know. I'm just a dude. I'm just Stijn. And I think if I can, across the vast expanse of this universe, in this very room, in this tiny speckle of time, transfer a bit of passion, a bit of joy, a bit of light to a couple of you, I think I've made an actual impact. And I think in there lies the Stijn signature. Thank you all for coming. This was a show. I hope you have learned something new, or at least enjoyed. I know this has been wildly inappropriate for a Monday afternoon talk about science. And perhaps a little bit weird. But even if you hated it, you can leave here knowing you brought some joy to this pretty weird, awkward, and often quite depressed little boy. and thank you so much for your attention. Yeah, so I don't know if there's any questions. This is a very blatant plug. I make YouTube videos. Go subscribe. I get 20 views, and each one of them makes me very happy.
So thank you very much.